is in the slideshow that you have, that I uploaded,
It's just the wrong name.
there's a typo.
Okay.
Yeah, it's not even just the wrong name, it's the wrong structure. If you look at what the slide has, it is the bisphosphate.
Oh,
But
okay.
that's the one that should.
But that's the one that should be there. You
Oh,
should
okay.
be starting with glyceraldehyde, not pyruvate, on whatever slide number that is.
Okay.
Right. So you want to copy that down now, you can copy that down. And this is the end of step 1, was the formation of our glyceraldehyde, getting everything to glyceraldehyde. Or step 1, the first part of glycolysis. It was to get to glyceraldehyde. Now that we're at glyceraldehyde, we need to convert that to pyruvate. Okay? And when we talked about this on Tuesday, we noticed a couple things. We were trying to identify big changes. So what were some of those big changes that we noticed?
Phosphate.
Okay? So we lose a phosphate. I'm just going to say lose P. Oops. Okay, what else changed?
Oxidation reaction.
Okay. We also have an oxidation reduction reaction because we tend to just focus on our organic molecule. It's not going to be called oxidation reduction. It's going to be called, in this case, oxidation, because we're going from the aldehyde just to the carboxylic acid. So we're really looking at just an oxidation here. You are right to do it. You also have to do a reduction, but it's where do we keep our focus? Okay. If we lose a phosphate, what does that potentially mean? What happens when we exchange phosphate? Energy, we're looking at ADP or ATP, depending on where we're going. So if we lose a phosphate, we're probably starting with ADP, and we're going to ATP. So this is useful. We're now looking at the formation of an energy molecule. That's good. If we look at the oxidation, that means something else would have to be reduced. So what is our good oxidizing agent or electron carrier? Is our NAD plus. If it is reduced, causing this to be oxidized, we would form NADH. So we now have an electron carrier. This could then later get used in oxidative phosphorylation, also known as the electron transport chain, okay, which will produce ATP. So that's cool. We've got something happening there. The other thing that's interesting to note is the ATP. We've now produced ATP. How much ATP did we produce?
2, 4.
So there's multiple answers we could do here. Okay, the first answer I would look for would be 2. Okay,
Just from this step is what you're talking about.
just from this step, just what we see. We have one phosphate, which would produce 1 ATP, but remember there's 2 glyceraldehydes because we split glucose in half, right? So that would suggest we could get 2 ATPs. But what are you remembering, Leanna?
You said 4 in your chart.
If we look at our chart, we're producing 4 ATP. To have 4 ATP, I need 2 more phosphates on here.
Oh, you said they're free. They're floating around. They weren't.
So we're missing those phosphates. We know at the end of glycolysis, there has to be 4 ATP. So somehow or another, we are missing, quote unquote, 2 phosphates. Okay? And so we could potentially put those phosphates onto our structure using ATP, but then we're going to end up in the same situation we were. Consume 4, produce 4. That doesn't do anything. Glycolysis does give us a net production of 2 ATP. So that means these missing 2 phosphate groups that we need to get us our 4 ATP cannot come from ATP. They have to come from some other phosphate source.
So where did they come from?
So then the question becomes, where do they come from? Remember, phosphate is relatively high concentration everywhere within your cell. Sometimes, if it's on one molecule, that molecule is now activated to react, and then very likely the phosphate gets kicked off, which then means in our cells, we have something that we can refer to as inorganic phosphate. Okay? Different from organic phosphate. Well, inorganic phosphate means there's no carbon attached to the phosphate. If we take a look at the phosphate in our glyceraldehyde, what do we have attached to it? Carbon. So that's organic phosphate. Inorganic phosphate would just be the phosphate structure floating around on its own. There's inorganic phosphate. So somehow or another, inorganic phosphate has to come into our reaction so that it can then be attached to our glyceraldehyde structure and then be removed later to produce our ATP. Okay, so now we're running into, okay, well, how can that happen? Well, we lost a phosphate, we did an oxidation reaction, There's a bunch of stuff happening here. Okay? And actually, I want to add one more thing. Okay, is that all that changed here? Loss of a phosphate and oxidation?
Did you gain hydrogen?
What's that?
Did you gain hydrogen? No, you didn't.
Let's label our carbonyl and our glyceraldehyde. Okay, so we have a carbonyl there. Where do we think that carbonyl is in pyruvate? It's actually the top carbonyl. Okay, that's the carbon that gets oxidized. Okay. So if that's the one that's doing the oxidation, and ADP is removing the phosphate, we lose that phosphate, what else changes?
The alcohol phosphate.
It's no longer an alcohol at one position, and so this disappears,
Oxygen also lost too.
Another one of those isomerization things.
and we end up forming the double bond, a second bond. So at our bottom carbon, which I'll star, what happened to that?
Hydrolyzed. What is or D? Or reduced, I guess.
So we could refer to it as reduced. It lost oxygen, gained hydrogen. So we could call that reduced, and we'll put it in quotes, or just red, because I'm lazy. Okay. What happened to the rest of the structure? What happened to that carbon right above it?
It was oxidized.
It was oxidized. What's the net result?
Nothing.
Nothing. Have we seen this happen before?
Yes.
Yes. And as Mike was already alluding to, we're potentially using an isomerase, okay, to shuttle that carbonyl from, or shuttle our alcohol, break that off and turn it into a carbonyl. Okay. So very similar to an isomerase type reaction. I'm not saying it is an isomerase, okay, but that same kind of concept is happening down there. So we have three major things happening here, which means we're looking at how many mechanistic steps?
3.
At least 3. Okay? That does assume that when we make ATP, that's happening in one step. That assumes that when we do the oxidation, that's happening in one step. And that the isomerization is happening in one step. Okay. We haven't even looked at the chemistry to decide if that's even possible. Okay? But that's where we're kind of keeping our focus. So minimally 3 steps. So let's go ahead and look at our very first step in the conversion process of glyceraldehyde to pyruvate.
So when the phosphate leaves, it leaves the oxygen?
How the phosphate leaves is a good point. It's a good question on how it breaks off. That's ultimately going to depend on how it reacts. Okay?
So on that one, it's going to leave the oxygen on?
It will end up leaving the oxygen on.
So it'll be different.
Yeah. So a different oxygen is going to have to cause the phosphate to break off. Okay. So our very first step, take a look at this. We got a crap ton of things going on here. What's happening in this very first step?
Just putting a phosphate on.
Okay, so we're putting a phosphate on. So that's all that happened?
No, because your NAD is getting engaged.
Because if we look at the phosphate, yeah, it's negative. It could go through and try and attack that carbonyl in a nucleophilic type addition. That would then break the carbonyl bond.
But you still have it over
Okay. But what happened to the hydrogen?
It jumps off.
It jumps off. Okay, jumps off as what? If I'm going to reform the carbonyl, that hydrogen's getting kicked off as what? Let's redraw this area. Our electrons come into the carbon. Carbon has too many electrons, so it kicks them up onto the oxygen. We form what's referred to as our tetrahedral intermediate, which is unstable. The electrons come back down to reform the pi bond. How many bonds would that carbon have? It would have 5 bonds. Red, the one we just made, the 2 blacks from the double bond, a black to the rest of our glyceraldehyde, and a black to the hydrogen. That's 5 total bonds. Can carbon have 5 bonds?
No.
No. That exceeds its octet. It has too many electrons, so it has to kick electrons out. Where are there electrons to kick out?
Hydrogen.
Okay. If we look at the net result, it has to be the hydrogen, which then means what is the hydrogen leaving as?
Hydride.
Hydride. It's taking the electrons with it. Okay. Is hydride a good leaving group?
No.
No. It is horrible. There's no way it wants to take those electrons. Okay. But it does have to happen. Okay. So we need something to collect that hydride. Okay. So good point. We have NAD+. NAD+ can grab that hydride to give us our NADH. Okay. In the course of that reaction, you'll notice that your phosphate still has that hydrogen attached to it. We'll end up losing that hydrogen to solution, and that's that H+ they're referencing out at the other end. Okay, so there's, we'll do some fancy labeling in here. There's that H. We'll make this a purple hydrogen. That purple hydrogen is now the H of our NADH. Okay? So that's the gist of what happens there. Okay, there's kind of a big issue here, though. What do we know about phosphate? It's happy. It is exceptionally stable due to resonance. Okay? The aldehyde functional group is partially reactive, but it's not super reactive. Okay? It's not a very good electrophile. Well, if that's not a very good electrophile, and the phosphate is not a good nucleophile, what are the odds the phosphate attacks? Not good. In fact, the reaction will not go. So how did we dodge this before? We bring in ATP to provide enough energy. But if we bring in ATP, we're not using a free phosphate, we don't have our energy surplus that we're hoping to get. So something else needs to be involved to help activate this reaction and allow it to occur. If we think back to that previous chapter, there were 4 things that we talked about: NADH, FADH2, ATP, and what was that last one? We referenced it as a generic thing first, and then we gave a very specific example. There's a lot of dead air gas.
Oh, the enzyme.
The coenzyme. Okay. And specifically, we looked at coenzyme A. And in coenzyme A, the big thing we've got is sulfur. Sulfur is a great nucleophile. Okay? And it also acts as a great leaving group. Okay? The sulfur will attack the carbonyl carbon, giving us our tetrahedral intermediate. Okay? The sulfur will facilitate the reformation of the pi bond and the kick out of the hydride. Okay? Because sulfur can stabilize that. That gets us our NAD plus converted to NADH. But now we've got a problem. Our glyceraldehyde is physically attached to the enzyme through a sulfur. Okay, well, now we don't have glyceraldehyde, we have glyceraldehyde protein. Okay, that's a problem. How do we get the protein separated from the glyceraldehyde? Well, remember, the sulfur generates an exceptionally good leaving group as well as a nucleophile. That generates such a high energy bond that what happens? Phosphate, being a weak nucleophile, okay, can now attack the newly oxidized carbonyl carbon. That will then eject our sulfur group, and we've done our full reaction. So the protein must supply a sulfur. Okay? One option is using coenzymes. That's a decent source of sulfur. There's another source of sulfur. Where's that other source of sulfur? Cysteine has sulfur. Same idea as coenzyme A. They both have a thiol group at the end, the SH. Okay? Cysteine, anybody remember it? Oh, cool, so I can get away with making it up. H3CO minus, I think is that. I know it has the thiol, but I don't know how many carbons it has. I think it's just one. Okay? So cysteine in the protein backbone will generate our sulfur. That sulfur, through protonation and deprotonation, okay, can become our active nucleophile, facilitating the reaction. So the chemistry of our sulfur is going to be super, super important, allowing a lot of this reaction to carry through. Did you find it?
Yeah, it just has one extra carbon.
Just one carbon? Cool.
So you have one more.
Oh, so there's two carbons in
Yeah,
the chain?
so
Ah,
there's CH2 and then goes to
that is a CH2.
SH. And then one more C to the SH.
Oh, there's another CH2. Fine.
Yeah.
Okay. Well, I can kind of understand it. A little bit longer it is, the further it can get away from the main protein chain. Fine. There it is. Okay. So what we're looking at is trying to acknowledge what things changed, figure out how that reaction happened. Okay. We looked at it just from our synthetic view, and the synthetic view says that ain't happening. The protein, and remember we talked about this, whenever we don't know how something happened, what do we say? The enzyme did it. What's happening? The enzyme did it. Okay. The specifics of what that enzyme is, or parts of that enzyme that facilitate it, that's where you start to come into kind of this gray area on how far into it do we really want to follow. Okay? Kind of makes sense. So it's looking a bit at the energetics of your reactions as well as kind of your overall big pictures. Questions? Okay, so let's answer the questions that we've got up at the top. Which happens first, the oxidation or the phosphorylation?
Oxidation.
The oxidation must happen first. The phosphorylation reaction is not a favorable reaction. We have to do the oxidation first. The oxidation is reacting with your cysteine. That's your big primary first step. Okay? Once it's now oxidized, we have a high enough energy that we can overcome the phosphorylation reaction. Make sense? Okay. So we're now at the 1,3-bisphosphoglycerate. Okay, and this is why it was such a massive typo to start with that, because that was not an easy thing to do. Okay, we produced a lot of kind of high energy pieces there. Okay, and a lot of that came from that cysteine bond. Okay, so we now have two phosphates on our structure. Okay, what changes in our next step? We got rid of a phosphate. Okay, so we might say, well, why the hell did we put it on to begin with? How do we get rid of it? What do we dump that phosphate onto? ADP to make ATP. We now have our high energy particle. That's why we did it. We're trying to generate our currency so that we can then go out and do our reactions using ATP. Okay, we transferred a phosphate group, which means, we transferred a phosphate group using ADP or ATP, which means we have a kinase. In that previous reaction, we did transfer a phosphate group. You'll notice the name is not kinase. Why not?
So it's cysteine.
We need ATP or ADP involved in the reaction for it to be called a kinase. Kinase will use your ATP, ADP. So regardless of the direction, those are your kinases. Here, we got the phosphate group on, but the only way we got the phosphate group on was by first oxidizing. Okay? What do we do when we oxidize something? We reduce it. What does reducing mean? Okay, it gains, wait, we're oxidizing, we're not reducing it. We are gaining oxygen bonds and losing hydrogen. It's another way to say lose. No, just general. You're playing a game, you guys run around playing a game, and you lost the game. What would you say? Whoa!
Didn't win.
Yeah, come on, keep going.
You have a sore foot.
Defeat.
Oh.
Loss. D. That's our point. D is our loss.
Oh.
And what did we lose?
Hydrogen.
Hydrogen. What's the name of our enzyme? A dehydrogenase.
Oh, very good.
Okay. Where did we lose our hydrogen from? Glyceraldehyde 3-phosphate. What's the name of our enzyme? Glyceraldehyde 3-phosphate. Losing hydrogen. Enzyme. Super creative. Okay. When we move to the next one, we're now producing ATP by doing a phosphate transfer from our bisphosphoglycerate to ADP. So we are now transferring a phosphate, and we are transferring it using ADP ATP as our shuttle, which then means we're going back to a kinase. Okay? So hence the enzyme name kinase.
So on all of these, though, we really haven't even looked at the enzymatic pathway, because like we're talking about everything that's happening, but we've never talked about what does phosphoglycerate kinase actually do, because we talked about what cysteine does, we talked about...
Well, what did we do here with the dehydrogenase? For this reaction to occur, what did we need?
We got rid of the hydrogen, but I thought cysteine was...
We needed cysteine to do that.
Cysteine in this...
Where is cysteine coming from? Cysteine is an amino acid. Amino acids are found in
The
proteins, and your enzyme
So that's where
is your
that's-
protein.
Okay,
The
gotcha.
cysteine
Okay.
is coming from your dehydrogenase. So there are certain steps where your enzyme is directly covalently linked to your substrates. This is a case where we get a direct covalent linkage between our substrate and the enzyme. Okay? And that's because this reaction is completely unfavorable. The enzyme must get directly involved. Because the reaction is like, I'm not doing it. F off. Okay? And so the enzyme has to come in and forcibly say, no, this is what's happening, do it. Okay? When we move over here, okay, forming ATP, well, that's a really high energy or endothermic reaction. That's not very favorable. But if we look at the energy of bisphosphoglycerate to phosphoglycerate, that is an exceptionally exothermic reaction. Okay? That reaction can potentially happen on its own. Okay? But you have to have the perfect alignment of all of those pieces. The first one and ADP. They need to be right next to each other to do that transfer of phosphate. Okay? So while it could theoretically happen on its own, what are the odds that those two things randomly are going to get close enough to each other to allow that reaction to occur? Really, really limited. So what does our enzyme do? It brings them together. Okay? So the kinase in this case probably isn't doing a covalent linkage to anything. All it's doing is bringing the two together and now saying, okay, phosphate, get the hell off of that one molecule. Okay? And by kicking it off of this one, it now has enough energy coming off of our bisphosphoglycerate that it gets launched into our ADP to then make our ATP. Okay, so that massive release of energy of kicking that out is probably what's shifting this along. How is it getting kicked out? Well, that's a good question. That's going to involve looking at all of those bonds, making and breaking. What are we using in each of those stages? Don't stress. Okay. We only really want to introduce the enzyme activity or its direct role when it's really, really important for it. Here, we've got enough of an energy balance between those that as long as the enzyme brings them close to each other, it's going to be catalyzing relatively simple proton transfers to allow the reaction to occur. The previous case, that ain't going to happen. The enzyme must get directly involved. So when the enzyme has direct involvement, then we're going to talk more specifically about what the enzyme is actually doing. Okay? Anytime where we don't get direct involvement, you just need to be you need to realize the name of the enzyme, unfortunately, okay, at least to some level, okay, and what the overall change is. Okay? You'll notice that magnesium is showing up in this case. Why is magnesium there? when proteins fold, we generate quaternary structure, or sorry, tertiary structure. What were some of the things that were important to folding a protein to get its final structure? Metal ions. Okay? The coordination of a metal ion within a proton or protein can help it fold properly. Why do we need magnesium? If magnesium's not there, the protein doesn't fold properly. If the protein doesn't fold properly, can it grab our starting materials? No. If it can't grab those, we don't facilitate the reaction. Okay? So we do need that metal ion to allow this to occur. Okay? There's this other statement that pops up here, substrate level phosphorylation. Probably should have mentioned this earlier as opposed to oxidative phosphorylation. Okay? Substrate level means we're just shuttling a phosphate, okay, to our ATP, okay, without having to do any transfer of electrons. So not redox. Oxidative phosphorylation means we're using reduction or oxidation to facilitate the phosphorylation. Like, for instance, the one before. Okay? We could refer to this. That's useful. What a dumb alert is that? Could be referred to as oxidative phosphorylation because we are doing an oxidation reaction, okay, to allow the phosphorylation to occur. Where your oxidative phosphorylation typically is being referenced is in the electron transport chain, okay, which is chapter 19, I think. That's one that I'm going to be surprised if we get to before the exam. So we'll just have to talk about that after the exam. Okay? So we've got our phosphate, we got our ATP. Okay? We did our oxidation and we generated NADH, but we're still missing another ATP. Okay? Because so far we've produced how much ATP? Careful. 2. How many glyceraldehydes did we start with? 2. Okay, so at this stage, we've now produced 2 ATP if we've fully reacted our glucose molecule. Okay, so what happens in the next step? Okay, we might say, oh, well, that's going to get us our phosphate. Okay, we can kick the phosphate off. Instead of kicking the phosphate off, what ends up happening? What's our change here? We move the phosphate from one position to another. Okay? Kind of like the reduction and oxidation reactions that we did partnered, where we generated isomers. We have isomers here, okay, but the isomerase is being reserved for noticing a reduction and oxidation reaction happening at the same time. Are we doing a reduction and oxidation reaction at the same time here? No. Okay. So in this case, all that's happened is we've moved the phosphate from one location to another. Okay, so we can't call this an isomerase even though those are isomers, because the name isomerase means something else. Okay, so what do they end up using for the enzyme name? Mutase, because we've mutated the structure by shifting stuff around. Effectively the same thing as an isomerase, but slightly different meanings in what's happening on how, or the relationship between those isomers. So in our mutase, we've just shuttled that over. Okay. And remember, when we talked about this, we said only 3 steps. We're now at 3 steps, and we haven't gotten our phosphate. Okay. We also don't have that other carbonyl. Right? Remember our very last step that we said needed to change? That last carbon needed to be a CH3. So why would we need to move the phosphate? Well, if the phosphate's there, that's going to be really hard to make that a CH3. Okay? So it's going to be easier if we can shuttle the phosphate to a different position. Why do we not shuttle the phosphate at an earlier time? Well, how big is phosphate? Really, really big. And if we look at where our phosphate was previously, those phosphates would bump up against each other. That becomes sterically problematic. Okay, so we have to get rid of one, and then we can focus on getting the other one primed and ready to run. Okay. Next step. Okay, what changed? So we lose the OH, and you're referencing making the double bond. Is that all that happened? What's the formula for water?
H2O.
If you get rid of OH, that's only one H. It's that H that was removed. What type of reaction did we do here? I know. This one you actually have the general form for, so you should be able to figure this one out. So we could start to say, well, we lost hydrogen, but we also lost oxygen. So that doesn't work. We start with one species, we end with two. Which reaction type starts with one and ends with two? Addition starts with two and ends with one. There's the word. It's an elimination. We eliminated a small molecule. We eliminated water from the structure. The result of an elimination reaction is the formation of a double bond. So we've done an elimination reaction here. Okay. So in this elimination, okay, we might now want to focus on coming up with an enzyme name. Well, the enzyme name we use here is enolase. Okay, well, that sounds a bit weird. Okay. What is an enolase likely going to do? Something involving an enol. I completely agree with that. What's an enol?
Yes.
And that is exactly what I was hoping to hear. What does an enol look like? An alcohol, yes.
And a negatively charged oxygen.
So that would be our enolate, which is fine. I can shift to that because I think that'll be easier to see later. So we're still on the oxygen. The ate or the all part. What's the ene part mean? Carbon-carbon double bond. This is now an enolate. Did I start with an enolate? Did I end with one? Where did I end with it? There's my enolate. Yes, the phosphate is still attached to it, which brings up an interesting point. If I get rid of the phosphate, do I have resonance stabilization there of the enolate? Absolutely. What did we just set up in prime? The very next reaction, which is going to do what? Eject the phosphate. Okay. So this is an enolase because I have made an enol. That's where the enolase is coming from. Okay. Questions on that? You guys don't seem happy with that, but I seemed happier about harder stuff, so that makes me kind of nervous.
That's good. Moving on, all right.
Okay, that works. So look what happens in the very next step. What do we get? ATP. Okay, why? Because we end up, if we eject that structure, or eject that phosphate, okay, and mechanistically our ADP comes in and attacks the phosphate, the oxygen stays with our phosphoenol. What would we end up forming? The enolate. And now we've officially got the enolate, we have resonance stabilization within that. Awesome, fantastic. Okay. But as Jimmy might come back and tell us, our enolate or enols are not particularly stable, and they will oscillate between two states, the enol state and the carbonyl state. The electrons will come in, forcing these electrons out to pick up an H+, and what do we end up forming? Okay. The reaction between these two is referred to as our keto-enol tautomerization. Unfortunately written enol-keto in this case. okay? Because we're oscillating between the keto and the enol form, the ketone versus the enol. Okay? Typically, when we look at this reaction, we start with the ketone because the ketones are easier to see, and then we move to the enol. In this case, all we're doing is running the reverse reaction. The keto form is always thermodynamically more stable than the enol form, which then means when we kick off that phosphate, now the structure says, hey, pop, I'm going to go over to the keto form and be nice and stable. And we end up with our pyruvate. We transferred phosphate using our ADP-ATP couple, which then means kinase. And again, we take a look, we're using a kinase. In this case, it's pyruvate kinase as opposed to phosphoglycerate kinase. Why do we use a different name? It's a different molecule. Okay, we're looking at reacting a different piece. Okay, you'll notice that it actually says phosphoglycerate kinase and not bisphosphoglycerate. Well, didn't we start with bisphosphoglycerate? All of our enzyme names have been, right, what we started with and then what happened. Okay. Why are we calling it phosphoglycerate and not bisphosphoglycerate?
Because it's named after the ending product that it makes.
It's always, or the way we've been looking at it, it's always starting with whatever we started with. But remember, your enzyme catalyzes both directions of the reaction. The name comes from how someone discovered it. How was it discovered? Doing the reverse of what actually is happening in the cell. Okay? I completely agree with your statement. I think it is incredibly stupid. Okay, but in fairness, is there a better way to name it?
Absolutely not.
I mean, you can think, if you think back to organic chemistry, and you think of all the naming rules that went into organic, that was a lot of rules for relatively simple structures. We're looking at an enzyme that is massive, okay? Probably thousands of amino acids, and each amino acid has, what are we looking at, 20 elements in it, 20 atoms? You're looking at 20,000 different atoms being arranged. It's going to suck to name that using organic nomenclature systems. Okay? So we did the best we could, which is come up with a kind of crappy nomenclature system, but it at least kind of applies. We're looking at either our product or our reactant, and then whatever the enzyme is potentially doing. Okay? And it doesn't work perfectly, but it's not bad. Okay? This gets us to the end of glycolysis. The end of glycolysis is pyruvate. We now have pyruvate. Okay, so we've done everything we can. Questions about all glycolysis?
What does PEP stand for?
PEP is phosphoenol pyruvate, which is that starting material. P for the phospho, E for the enol, P for the pyruvate. Right, just like organic chemists, we get lazy, we don't want to write out the full name, so we come up with some stupid acronym, which then turns into alphabet soup, but that's what happens. When it comes to an exam, if I use an acronym and you don't recognize the acronym, raise your hand and be like, hey, what did this mean? Some cases I might tell you, some cases I may not. Like ATP, if you ask me what ATP is, I might be like, no. PEP, I'd probably tell you PEP. Right. To a certain extent, does the name help? Like if I came up and told you ATP was adenosine triphosphate, you go, oh, that makes things so much clearer. Right. So it depends on how much you've memorized and where you memorize things. Okay. I would say the same for the reverse, because if you go through and memorize PEP as opposed to phosphoenol pyruvate, and I write phosphoenol pyruvate, you're like, is there an acronym for this? Fine. Go ahead and ask. And again, I might tell you, I might not. But it doesn't hurt to ask. Okay? After that, we run into some kind of control points. We already talked about some of these. Okay? In the initial phases, we had control points wherever we used ATP. Why might we want control points wherever we used ATP?
Don't want to overproduce it.
We don't want to overproduce. If we start glycolysis and we crank all the way through that, we're producing energy. If we have a ton of energy already, don't start glycolysis. Okay? So we will cut off those steps. Okay? So those are some big control points wherever we've got ATP being involved. Okay? You'll notice that we get that again down at the bottom as well. The phosphoenolpyruvate is another control point. Interestingly enough, we don't see a control point at the glyceraldehyde to the bisphosphoglycerate. Yeah, that's the one. Okay, that's not a control point per se. One of the things that is important, though, is we need to have what to do that? NAD+. Okay, NAD+, because we're converting it to NADH. Okay, so if we don't have any NAD+ floating around, well, that reaction can't run on its own anyway. Okay, so that might be why that isn't necessarily a control point, because it's a relatively quick and easy reaction to run. Oh, and actually, does it involve ATP?
Just the floating.
It's a floating phosphate. It's an inorganic phosphate. There is no ATP there. So sure, fine. Take a phosphate from the solution. Who cares? That doesn't give me a whole bunch of energy currency floating around later. So it's not as big of a deal for a clutch point. Control point. There it is. Okay. So it is going to be important to memorize those enzymes, because those enzymes are your kind of clutch points or your control points. So you should go back and reference those and make sure you know that what is happening in each of those stages. I'm not a big fan of the memorization, so more than likely what I would do is draw out nasty reactions and say, here are some reactions. Which of these could be control points in glycolysis? If you see ATP, that's a control point. You don't see ATP, that's not as big a control point. Okay? The next part is anaerobic metabolism of pyruvate. So once we've gone through and produced pyruvate, what happens if we don't have oxygen? Okay? Because if we have oxygen, this is now the Krebs cycle, which is the next chapter. So what they're going to focus on here real quickly is just, okay, we don't have oxygen, what happens? Okay? Well, what we're going to end up doing is converting that across into lactate or lactic acid. So when your muscles start to burn, why are they burning? Buildup of lactic acid. Why is there a buildup of lactic acid? Because you haven't been breathing in enough oxygen to get to your muscles for them to run under aerobic conditions, and we shifted to anaerobic, which then gets us the lactic acid buildup. Okay? So why go through this process? Well, we end up generating NAD+. Okay, the NAD+ can then be used later to pick up NADH or do your oxidation of glycolysis to help produce more energy. Okay? Because if we don't go through the Krebs cycle, we need to get more NADH. Okay? Well, glycolysis produces some NADH, okay? And that NADH can go into oxidative phosphorylation. Okay? But if we can't get anything out of the Krebs cycle, we're going to need to start producing more NAD+ so that we can then do glycolysis to get more ATP. So we end up then relying on our ATP production solely in glycolysis. Well, the issue for glycolysis is the presence of NAD+. If I have NAD+, I can then move through and produce ATP. Do you guys remember that?
Right.
Kind of sort of connect? Okay. So if we can't produce ATP, okay, through our NADH couple with our oxidative phosphorylation, we'll instead take that NADH and we'll double it back into glycolysis and say, okay, produce ATP solely out of glycolysis. Okay? And that control factor is then the NAD+. Because to do the glycolysis, you need NAD+. Okay? I wish I had I don't think I've got a picture for this. That doesn't do what I want. That doesn't do what I want it to do. I'll try and come up with a better drawing for that for Tuesday so that you can see that connection. This ends up allowing us to generate more ATP in glycolysis because we now have NAD+.
And that can float back up,
Go back into glycolysis, then allowing glycolysis to produce 2 ATP. It's a bad way to produce energy because we're now only getting 2 ATP. Whereas if we take NADH and dump it into oxidative phosphorylation, we get a lot more ATP, but that requires oxygen. There's no oxygen. All you're doing is building up NADH that can't do anything. Okay? So to get energy, we'll take that NADH and we're going to burn the energy pretty much. Okay? We're going to say, I can't use that to produce ATP reasonably, so what I'm going to do is kill it, refresh my NAD+. My NAD+ can now go back into glycolysis to produce 2 ATP. Kind of, sort of? Okay. This has another tetramer molecule, so they mentioned that. You'll get heart and muscle subunits. So you'll find lactic acid or find different combinations of that tetramer in different locations. So if you look at heart tissue, which is a muscle, okay, you will find the H subunit. Okay, you'll find primarily H4 in the heart. There's 4 of those heart subunits. Okay. If we move to the muscle, you'll find 4 of the muscle. Why do we want a different process in each of those? Yeah, go ahead. You don't want what?
Lactic acid building up in your heart.
Lactic acid in your heart, generally not a good idea. Okay? If we have to absolutely generate energy, the heart needs to be able to do it. Okay? So we will have some kind of control there. We're going to have a slightly different unit because I don't want this to be the go-to mechanism, okay, to produce ATP. But if I gotta, I gotta. Okay? And that's why we have those 2 separate types of units. Depending at where you are within the body, you'll end up with varying amounts of each of those subunits.
So you said it's a tetramer. Is it a pyruvate tetramer? Like
The tetramer goes back to the protein,
So it's just we're not looking at the whole protein itself.
like hemoglobin.
Okay, so it's
So that tetramer is referencing it's like hemoglobin, okay, where it can then pick up oxygen. So that tetramer can now pick up pyruvate and NADH and allow the reaction to occur.
The enzyme?
The enzyme is a tetramer, and it's a dehydrogenase. Why would we call it a dehydrogenase?
Well, it's taking it off the NADH.
So we again run into this question of referencing on where it's going. A dehydrogenase is going to be removing hydrogen. Okay, defeat. Okay, but we're calling it lactate dehydrogenase. But aren't we starting with pyruvate?
yes, but lactate was
When it was initially discovered, they noticed the process of going from lactate to pyruvate was what they tested. What happens is you go from lactate to pyruvate. You lose hydrogen. That's why it's lactate dehydrogenase. Okay, because they discovered it going lactate to pyruvate, even though pyruvate to lactate is how it would function in the body. Okay?
So the H and the M4 refer to the 4 different units on the enzyme? Or there's
Yes, 4 different units within the enzyme. Okay, that would be its quaternary structure, and not quaternary because there's 4, but quaternary because there's multiple different subunits.
Those 4 points can all react.
That's a good question. I don't know that. Okay. And so this is saying the same thing that I did before. NAD plus needs to be recycled to prevent a decrease in your oxidation reactions. So really what we're saying is we need ATP. How do we make it? We need NAD plus. Okay. So that's why we're going through this process. Okay, we need more NAD plus. They've gone through and shown 2 different ways to do this. Lactic acid fermentation, okay, which is what happens in mammals, I think, exclusively. And then alcoholic fermentation. Alcoholic fermentation happening in bacteria and yeast. they go through and process sugar, and when we cut off oxygen supply, what do they turn it into? Alcohol, which we've then used as humans to make our lives so much easier, or much, 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 much harder. Okay? So you're using a similar process in each case, just different end results. Okay? Questions? Okay. If we look at the alcoholic fermentation, they mentioned the decarboxylase, okay, which we'll end up talking about again a little bit later, okay, where it's relevant within Krebs cycle. Okay. But if we take a look at what's happening as we go from pyruvate to acetaldehyde, what changed?
Carbon dioxide.
Okay. CO2 was formed, which came from my nice little red box. Okay. So what did we do? We removed, or a carboxylate, okay, which is the CO2. So we decarboxylated. Okay. What if we wanted to look at acetaldehyde to pyruvate? Okay, well, then we might call that acetaldehyde carboxylase. Okay, but again, depending on how we looked at it or how it was initially discovered, we haven't discovered this pyruvate to acetaldehyde. The enzyme name gets associated with the initial discovery. It's pyruvate decarboxylase because we're removing a carboxylate from the pyruvate. How that happens is absolutely fascinating. Okay? So, requires magnesium, and it requires this cofactor called thiamine pyrophosphate. Okay? Super, super important to metabolism. We need that cofactor. Okay? How it works, I don't think is this next slide. I lied. We've got thiamine, which is vitamin B1. Okay? The big chemistry all comes into play with our inner ring, in particular that sulfur. What did we say was special about the sulfur? It's a good leaving group and a good nucleophile. It can attack the carbonyl carbon of this. Okay. Through a sequence, actually it's not the sulfur, it's the carbon that ends up acting. Okay. Which seems a little bit weird because does the carbon really have any electrons? No. It turns out that that carbon, and in particular the hydrogen, attached to that carbon, is actually quite acidic. Meaning I can remove those electrons and make this a negative charge. This becomes even more fascinating because I can take those pi electrons and put them on the positively charged nitrogen. Which then means what is interesting about that carbon? Number one, it has extra electrons. Number two, if I take those pi electrons and put them on the nitrogen, the carbon is missing electrons. I have a single atom that can act both as an electron donor and an electron acceptor. Right? This is made even more interesting because it's carbon. It is so interesting that in organic chemistry it is referred to as a, and I doubt even Jimmy remembers this, a carbene. Okay? The hybridization of that carbon is just so bizarre that it gets its own special name. Incredibly reactive species in the real world, okay, or in organic chemistry. In biochem, the thiamine molecule can stabilize that carbene structure well enough that it doesn't just destroy shit in our body. Okay? It can help turn that carbon as a good nucleophile and as a good electron acceptor, facilitating the elimination of that CO2. Because if we look at what happens for the CO2 to get eliminated, the electrons have to come in, those electrons have to go to that carbon. Can carbon stabilize a negative charge? No. How does it stabilize it? Through a linkage with the thiamine. Okay. I don't know if I've got it. Yeah. Okay, so if we go through, we generate the nucleophilic carbon, which can attack our carbonyl. Okay, and this one, unfortunately, is not showing the best system. That can polarize it. We can end up doing our elimination. Okay, our CO2, our oxygen from our CO2 can come in, form a double bond with our carbon, but then we end up breaking that carbon-carbon bond. Those electrons can go into the thymine structure. Okay. The electrons in our thymine can then come back down, pick up some acids, or pick up a hydrogen from an acid, and we can continue that process around to then kick it back out where we have our carbonyl compound and whatever our ejected molecule was. Okay. I don't want to go into it in too much more detail because I know we're going to talk about it again, I think within the next 5 or 6 slides, where I think I've broken it down a little bit cleaner, okay, than this. Okay? So it is a fascinating molecule. The big things that I will expect you to know, thymine and carbene, that's a pretty important terminology to tie together. Okay? Within that, your carbon acts as both a nucleophile and an electrophile, okay, of your thymine molecule. End result, glycolysis beginning to end, releases 73 kilojoules per mole of glucose out into the world. That 73.4 kilojoules per mole happens to correspond to the same amount of energy necessary to make 2 equivalent moles of ATP. Your ADP to ATP is 31 kilojoules per mole. That's not right, I can't do the math. 36 kilojoules per mole. So your 73 is 2 of those, so that allows for that. So your big result is that you get 2 ATP and 2 moles of pyruvate. And that 2 moles of pyruvate becomes important because now each of those pyruvates moves through the Krebs cycle. So when we talk about the yields of all of our energy carriers in the Krebs cycle, you have to realize you have to multiply those now by 2 because there's 2 pyruvates moving into it. Okay? That was chapter 18. Hooray! Okay. So chapter 19, citric acid cycle. If we look at our glycolysis, we went through and did really 2 big things. Okay, if we skip all the way back to our, I'm going to regret doing this. If we look at our glycolysis, we went from glucose to fructose 1,6-bisphosphate, or glucose to glyceraldehyde. That was pretty much stage 1. Okay, and when I accidentally had my wife try and talk me through some of these reactions, she started using terms that I was like, I don't want to use that. I wanted more chemistry. But having then thought about it, it actually makes sense. That first stage can be referred to as the investment. Because what's happening is we go from glucose to the glyceraldehyde. We put the energy in. Okay? That is our energy investment. Okay? In the second stage, we're getting our return. And in our return, so in our investment, we lose 2 ATP. Whoops, I meant to put a 2 there. And in our return, we gain 4 ATP, and we gain 1 NADH. Okay, so we really had 2 major phases within glycolysis. Each of the steps within that use different enzymes, use different properties to ultimately set it up and allow for the return. It's really all they were there for. Shuttle things around so that we could then break the structure apart, that we could then put phosphates on, that we could get that return back out of it. Okay? And if we look at that total, we got 1, 2, 3, that's 3, 4, 5, 6, 7, 8 9 reactions. Okay. Once we move into the Krebs cycle, see if I can actually get to the slide I wanted, we're now adding a bunch more reactions on top of this. We're going to end up with 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 reactions. So all the reactions we had in glycolysis, we're now doubling those reactions by looking at the Krebs cycle. Okay? So lots and lots of different reactions that you need to be focusing on and seeing what's happening. Okay, before we get into that, our big picture. Our glycolysis produces the pyruvate. Pyruvate then dumps into, seemingly in this figure, acetyl-CoA, okay, which makes it look like it's this big, massive reaction. It is and it isn't. Okay? It's a massively important reaction that is involved in a variety of different chemical pathways, but it is really only one massive reaction. Okay, so it's one enzyme that's doing that. The product of that gets dumped into the citric acid cycle. The citric acid cycle, as the name suggests, circles back on itself. So the product of the end of the citric acid cycle ends up being the reactant to start it. Okay? And in that cycle, we end up shuttling electrons around to the point where we can make a whole bunch of electron carriers, primarily our NADH. Okay? FADH2 is a little bit weird to mention because it's not mobile, okay? But it is technically getting made and converted into NADH. Those electron carriers, we can then carefully react with oxygen to reduce oxygen. Well, that reaction is highly exothermic, which can give us enough energy to produce our ATP. Okay? So what we're ultimately trying to do is get all the way down. All of this process is really just for this reaction at the very end. That's it. Okay? It's just a question of how we get there. Do we want the control points? Well, yeah. If we don't have the control points, then if something goes wrong, say with a single enzyme that processes it, then we're SOL. Okay? Maybe each of those little pieces could then be used to produce something else. So if we can intricately link all of these things, we can then minimize local fluctuations within our cell concentrations of any of those reagents, and then we can kind of maintain the production of energy without losing anything, okay, or losing minimally. So how do we put it all together? Yeah. That then becomes our citric acid cycle. I kind of like this figure because it's pretty. I also kind of don't like this figure because it has a lot of colors and it just kind of gives me a headache. Okay? But it does at least show everything. You're getting names, you're getting enzymes, you're getting products coming out, species going in. It gives you a lot of information. Okay? I don't like how the textbook, and in fact, I'm not even sure I have what the textbook organized as a figure, because theirs I really didn't like. But this at least puts the entire chapter, I already forgot what chapter it is, 19, on one slide. This is it. You're responsible for all of that. Okay? What we will proceed to work through Tuesday and for 5 minutes today, okay, is looking at some of those reactions and saying, okay, what's happening? Just like we did with glycolysis. How are the structures changing? Okay? At each of those stages, do we produce energy? Do we need energy? How is that going to work? Does that make sense? Okay. To start this all off, we have to pick up with where glycolysis ended. Glycolysis ended with pyruvate. Pyruvate's right there at the top. Okay. The very first step is to convert pyruvate into acetyl-CoA. So we went through all of that glycolysis stuff to really boil it down to an acetyl group. That's all I wanted. Okay? And that group is present in pyruvate. And I know it's not super obvious, but I can circle it in purple there. Okay. So what's the part that I don't want? Well, that looks an awful lot like 1 carbon and 2 oxygens, also known as carbon dioxide. So one of our processes of respiration is to release carbon dioxide. Why? I took all of the glucose and I boiled it down to really only wanting 4 total carbons of glucose. The other 2 carbons I don't want, I'm ejecting those as waste. That's my CO2. So if we actually consider that's actually pretty phenomenal. We started with 6 carbons and we're using 4 of those to produce energy. 2 of those we're excreting as waste. That's actually not too bad. Okay. So we need to get that acetyl group. So kicking that CO2 off is not the easiest task. That's where we bring in pyruvate dehydrogenase. Okay. This dehydrogenase is going to remove hydrogen and CO2. Okay. So the question then becomes is how does it do it? Okay. It involves a bunch of stuff because you can see there's a bunch of stuff crammed in there. Okay. One of the big things to note, see NAD+, we're generating an electron carrier. So this first step is going to get us an electron carrier, which we can then later move to oxidative phosphorylation, our electron transport chain. Okay. To get that, though, I have to use coenzyme A. Okay. So coenzyme A is now being intricately linked into this process to allow me to run through this reaction. Okay. So the pyruvate dehydrogenase needs to bring in NAD and coenzyme A and pyruvate and get them all to work. Okay. Interestingly enough, it's not that simple. That's what this slide is trying to show. We start with our pyruvate. Pyruvate reacts with the enzyme. Okay? And in particular, what molecule is it reacting at within the enzyme? Our thiamine pyrophosphate, which was vitamin B1, which actually is not sulfur. Remember, that was our carbene compound. Sorry, it's not drawing. Okay, there it goes. This was our carbene compound. Is that nitrogen? Okay. This carbon was now our good nucleophile. That carbon comes in and attacks the carbonyl of our acetyl group. Hey, that's what we're trying to get. That facilitates, through a whole bunch of mechanistic arrows, which we already kind of talked about, the kind of consumption of the oxygen's electrons into the structure, which then shuttle electrons out. So we end up ejecting CO2. So there's the loss of CO2. As a necessity, though, our acetyl group is now attached to TPP through the carbon that we circled earlier, that blue carbene carbon up at the top. Okay. Well, I don't want that structure. I need the acetyl. And right now, I have an alcohol. So what do I have to do? I'm going to have to react that with something else. What does that something else need to do? Remove the hydrogens. That something else needs to do what to our acetyl group? It has to oxidize it. Okay. This is where our protein comes into play with lipoic acid. Okay. Lipoic acid happens to have these disulfide bonds. Disulfide bonds are fascinating because they allow for or facilitate redox reactions really, really quickly back and forth. Okay. So the lipoic acid will help the oxidation. So, and by doing that oxidation, you'll notice that we take our acetyl piece and transfer it from TPP to now being attached to our lipoic acid within our protein. Okay. The whole point was to get the acetyl group actually to coenzyme A, because coenzyme A is now mobile and can transfer that acetyl group through a high energy sulfur bond Wah! Why'd you do that, stupid computer? Remember, this is our high energy sulfur. That if I can get the acetyl group out onto a mobile structure, like acetyl-CoA, okay, this is now mobile, I can then get that reagent to the next step in my reaction. Okay. So we start with pyruvate reacting with our thiamine. The thiamine structure then transfers that, for lack of a better term, our acetyl, to our lipoic acid, while at the same time oxidizing it, so we now have the acetyl. The acetyl group, you'll notice as well, also has that high energy sulfur bond. Why use coenzyme A? Well, lipoic acid is bound to the enzyme. That's not very mobile. Okay. Why would I want to bind it to the enzyme? It's really good at oxidizing and reducing. I want to control that oxidation reduction reaction. I want it to happen only in this site, not anywhere else. Okay. I already have a good electron carrier in NADH. Okay. So we end up transferring the acetyl group from one high energy sulfur bond to a second high energy sulfur bond, but that second high energy sulfur bond is now mobile and can now piss off into the world and do whatever it needs to do. Okay. Meanwhile, if we think back to what happened to our enzyme, okay, at each of the stages within that process, thiamine pyrophosphate then became attached to our acetyl group. Well, that's bad because it's no longer thiamine pyrophosphate. The enzyme's dead. What does it end up doing? Well, it transfers that acetyl group to something else, which then refreshes our thiamine pyrophosphate. So it's now active again. But by transferring it, we've now killed the lipoic acid. So now the lipoic acid is non-functional, and our enzyme is still not able to continue its process. Okay? But then we go through and shuttle the acetyl group off to coenzyme A. So now I still potentially have my free sulfurs, but the issue is to get them from the free sulfurs to the lipoic acid. What do I have to do? I have to remove two hydrogens. I have to do an oxidation reaction on them. Okay, they must be oxidized. So I need something else to be reduced. What's that something else? On this slide, they make it look like NAD plus is being reduced. But you'll notice it's not just NAD plus. What are they also showing there? FAD. Why are they showing it that way? Because they probably ran out of space and didn't want to draw anymore. It actually gets reduced or oxidized by FAD. FAD is then reduced, but FAD is not mobile. So what does the FAD H2 then do? Dumps the electrons onto the NADH or onto NAD plus that can then go out as a mobile NADH. Holy crap. And all of that for 2 carbons and an oxygen.
So in that picture, the sulfurs on the lipoic acid, those are turning with the cycle, it's not...
So a good question there would be, are there actually 3 of them there, or is that just their way of showing each of those enzymatic steps? And I think it's they're just showing.
Like it's turning with it.
Yeah, and I don't.
That was confusing when it first
think,
started. I was like, what is going on?
I'm not sure that it's actually turning with it. I'd have to take a look at that. I think they're using that to demonstrate the structural changes. Okay. So after all of that, we eject our coenzyme A, our now acetyl coenzyme A, because it has an acetyl group attached to it. We've ejected NADH, and we're now right back to where we started with our enzyme ready to go and do it again. Okay. Okay. Here's our thiamine being really cool. This type of reaction from OChem produces, actually, let's see, did I draw it? I didn't draw it. It's kind of neat that if we ran that reaction up top, what we'd end up with would be this structure. That carbon, which came all the way back from over here, okay, ends up acting as both a nucleophile and as both a nucleophile and an electrophile. The base removes the hydrogen. The carbon now has electrons. Bromine is a great leaving group. Bromine can ditch the structure, and so our carbon becomes a lone pair of electrons and an empty p orbital. That empty p orbital can act as our electron acceptor, and the lone pair of electrons can act as an electron donor. Thymine's doing the same thing, okay, through a slightly different system. Okay? If we look at organic chemistry, that carbene is stupidly reactive, okay, not stable remotely in the slightest. Thiamine pyrophosphate is actually relatively stable, so we can control when we want that active nucleophile to be there by either putting the hydrogen on or taking it off. Okay. We will pick up there on Tuesday. Let's see, do I actually go into it in more detail? Yeah, we'll walk through this mechanism. So what I'd recommend you do is take a look at this mechanism over the weekend and see if you can come up with any questions about it, and then we'll talk about it on Tuesday before we inch our way into the rest of the Krebs cycle. You'll notice periodically throughout this